So amino acids. So ammonia is a compound of nitrogen and hydrogen with the formula NH3. And amino acids is going to be used very, this is used a lot in the genetic material makeup of humans and so on. And we're going to go over this, and then this will help throughout the whole video. So amino acids. Ammonia is a compound of nitrogen and hydrogen with a formula NH3. It is ammonia. Or actually, better yet, I'll just put it over here and just edit it about a bit. So it is a colorless gas with a distinct pungent or strong smell. So and it's like this. So this is the ammonia right here, and H, and then there's an H here. These indicate just a different three-dimensional, like how it looks three-dimensionally. So anyways, I believe this in the background. This is more in the forward here, and then these two dots like that. So this is a NH3, so it's 3 H's, and there's an N. And the 2 dots are Lewis dots and represent a lone pair of electrons. So there's a lone pair, like that. So yeah, so there's lots of ways of, and lots of different ways people write chemical formulas or chemical structures and so on. So amines are compounds and functional groups. Remember, functional groups have the characteristic, or they're responsible for the characteristic properties of certain molecules and so on. They always react almost regardless of the rest of the molecules in that compound. So amines are compounds and functional groups that contain a basic nitrogen atom with a lone pair. So lone pair, basic like that. Amines are formally, this just has it by itself with a lone pair. Contain a basic nitrogen atom. Yeah, with a lone pair of electrons. So amines are formally derivatives of ammonia, so derivatives of this, wherein one or more hydrogen atoms have been replaced by substitution such as an alkyl or aryl group. So replace one of these, and now you have an amine. These may respectively be called alkylamines or arylamines, and amines in which both types of substitutions are attached to one nitrogen atom may be called alkyl-arylamines. All right. Important amines include amino acids. Yeah, so that's what we're getting to. So these are, it's all derived from ammonia, and ammonia amines derived from ammonia, and then important amines are amino acids. And the substitution NH2 is called an amino group. So when you add this one here, you're adding an amino group like that. And going further, the types of amino groups. So primary 1 degree, and then secondary 2 degree, and tertiary or 3rd degree amine basically has the number of hydrogens removing from this ammonia. So you have ammonia 3 hydrogens. So if you have primary amine, you remove 1 with the R group. So that's called amino group. And then if you write, and this one's called imino group, is when you replace 1, 2. And if you replace all 3, you have a nitrilyl group. So again, just a bunch of labeling. All right, so let's go further. So amino acids are organic compounds that contain, then I put this in bracket, charged amino, just to be more accurate, which is -NH3 plus. And here's a star I put in, and then this is from Wikipedia as well. Strictly ammonio as amino refers to uncharged -NH2. So then I just made it a charge by adding another H, by adding another 3 there. Or actually, when you charge this one, if you add another H, that would be just another plus. So basically, then you'll have this H3 plus there. So, but this term is almost never used. Yeah, so this is strictly ammonio, but because amino refers to this, but they'll just call it that. So I just put this charge there, and also put this other bracket here, or -NH2 plus in the case of proline, another amino acid. Yeah, so it contains NH3 plus or NH2 plus, or possibly any other similar ones, and carboxylate, which is CO2. Yeah, this is minus -CO2 and a negative charge out of the minus. Functional groups, along with the side chain R, specific to each amino acid. Going further, so the elements present in every amino acid are carbon C, hydrogen H, oxygen O, and nitrogen N. In addition, sulfur S is present in the side chains of cysteine, yeah, and methionine and selenium, Se, and the less common amino acid, yeah, selenocysteine. And you have here's a structure of a generic L-amino acid in the quote neutral form needed for defining a systematic name, without implying that this form actually exists in indetectable amounts, either in aqueous solution or in the solid state. So yes, it's just basically used for naming purposes, but here's a generic structure. So there's the N, there's the H, and there's another H there, and there's the R. And there is the CO2 over here. This would be a carbon, the C, whenever the dash line, it's often referring just to a carbon. The carbon chain CO2, yeah, and so on. And also this L, the reason for the L is because of a phenomenon called chirality, or handedness. So just to illustrate that here, so here's a figure here, D and L configurations of a chiral or handed amino acid. R is the side chain of the amino acid. So the L side here, we have this H2N on this side, and it's a mirror image is on this other side. So this is an L amino acid, this is a D amino acid, indicating it's a mirror image of itself. And so this one here is just a different form of the exact same setup here, but this one's rotated. Notice that the COOH is over at the top, and then if you scroll up, and then if you scroll up here, this one's at the side, so you just got to rotate this over to here, then you'll see it over here, COOH. H is going to be over there. Alright, so yeah, so that one's going to be like that. So the same setup, just different forms indicating. So one, yeah, just indicating, well, yeah, just the different types of writing chemical formulas. Anyway, so now this is the mirror image, and then going further on the topic of chirality. Yeah, so a molecule or ion is called chiral if it cannot be superimposed onto its mirror image. In other words, just placed directly on top and be exact same by any combination of rotations, translations, and some conformal changes. This geometric property is called chirality. The terms are derived from ancient Greek here for the word hand, which is, yeah, this is care or chair, which is the canonical or standard example of an object with this property. In other words, yeah, your hands are chiral of each other. And here's just your hand, your left hand, your right hand, or if you look in the mirror, your right hand becomes your left, and you can't superimpose your left and right hands together. So a chiral molecule or ion exists in two stereoisomers, which have, in other words, this means they're the same molecular formula, sequence of bonded atoms, but different. Let's write a but here. But different 3D orientations of their atoms that are mirror images of each other, called or I think it's pronounced enantiomers. Yeah, so called enantiomers. They are distinguished as either right-handed or left-handed by their absolute configuration or some other criterion. The two enantiomers have the same chemical properties except when reacting with other chiral compounds. They also have the same physical properties except that they often have opposite optical activities, which is this is ability to rotate or rotation of linear polarized light. Or right here, ability to rotate linear polarized light. So yes, very interesting. So here is an example, and they also amino acids a good example of this. So two enantiomers of a generic amino acid that are chirals. Right hand side, here's the left-handed side, and this one here, yeah, this one, the one that we're looking at over, this one here is NH2 is on this other side, on the left side, that is over here. This is an L-amino acid, this is a D amino acid. It's interesting stuff. Let's continue further. So peptides. So peptides are from Greek. From Greek language over here. Peptos, for meaning digested, are short chains of amino acids linked by peptide bonds, which is a type of a covalent bond involves release of a water molecule. So yes, it's covalent bond and releases water. So here's an amino acid 1 here. Here's an amino acid 2 here. This is a peptide bond formation via dehydration reaction. In other words, a loss of water from a reacting molecule or ion. So here's what we have over here. So then these link up. Yeah, and there is the water molecule. Well, there's an OH and there's an H. So then you're going to get an H2O gone. You're going to link up here. And then this end connects to this one right here. So here's a peptide bond. This forms a peptide bond over there. So a connection, and then it removes the water over there, and then you got a dipeptide, which is two peptides together. So this is amino acid, and there's amino acid 2, and these are again just short change of amino acids. So you have two amino acids, that's just two peptides, so dipeptide or two peptide. And going further, so an oligopeptide, often just called peptide, and oligo means a few. or quote, a few, consists of 2 to 20 amino acids and can include dipeptides, tripeptides, tetrapeptides, and pentapeptides. There's 2, 3, 4, and 5 peptides or amino acid chains together. A polypeptide is a longer, continuous, unbranched peptide chain. Again, it's just all terminology, because even this is a polypeptide because it's multiple, but it's specifically discussing shorter types. So a longer unbranched unbranched peptide chain. A polypeptide that contains more than approximately 50 amino acids is known as a protein. So protein is just a bunch of amino acids combined together. So there's amino acid. Has that ammonia or charged amino plus carboxylate. Or go back here. Yeah, so carboxylate. Functional groups along with a side chain R. So that's what it has. And then if you link up a whole bunch of those together, you get peptide and a lot more. If you link up a couple, you get a peptide, dipeptide, and you link more, you get a oligopeptide, or even more, you get a polypeptide, and then you get even further. That's called a protein. That's very interesting. So proteins consist of one or more polypeptides arranged in a biologically functional way. So it actually has a biological function together. Often bound to ligands such as coenzymes and cofactors, or to other protein or other macromolecules, or other macromolecules such as DNA or RNA. Yeah, so a bunch of amino acids connected to other macromolecules, such as, again, DNA, RNA, or to complex macromolecular assemblies, whole bunch together. So a ligand, so this one here is, so protein is often bound to ligands, such as coenzymes, cofactors. A ligand is a substance that forms a complex with a biomolecule to serve a biological purpose. The etymology stems from Latin ligare, which means to bind. And a coordination complex, this is short for complex. Complex refers to this coordination complex. So a ligand is a substance that forms a complex with a biomolecule and serves a biological function. So a coordination complex consists of a central atom or ion, which is usually metallic and is called the coordination center, and a surrounding array of bound molecules or ions that are in turn known as ligands or complexing agents. So here's a central one here, and a whole bunch of here ligands here. Ligand is a substance that forms a complex with it. Yeah, so then it all forms connected to it. Yeah, so it's ligands are complexing agents, so they're making a complex of this atom, which is it's usually metallic or a metallic ion, or a metallic atom, such as iron and so on. In this case here, cisplatin, which is PTSL2-NH3. 2 is a coordination complex of platinum 2 with 2 chloride and 2 ammonia ligands. So yes, you have 2 chlorides, assume these ones are it, and then you have ammonia ligands over here. Alright, let's look up the term for now, cofactor. So again, cofactor, right here, often bound to ligands such as coenzymes and cofactors. It's also a ligand. And it's so a cofactor is a non-protein chemical compound or metallic ion that is required for an enzyme's activity as a catalyst, and a catalyst is a substance that increases the rate of a chemical reaction. So cofactors can be divided into 2 types: inorganic ions, which lack a CH, carbon hydrogen bonds, and complex organic molecules called coenzymes. So coenzymes are more complicated here. Which can include CH bonds right here, and then this inorganic ones don't include CH bonds. So polypeptides and proteins are often referred to being comprised of amino acid residues. So you'll hear this a lot. And a residue is whatever that remains after a series of events. So in molecular biology, a residue refers to a specific monomer within a polymeric chain of a polysaccharide, protein, or nucleic acid, which has some discarded atoms during a condensation reaction. Where you're combining 2 or more into into less molecules, or one molecule. So the concept of let's fix this up. The concept that suggested this term is presumably the nature of the condensation reaction, by which such classes of monomeric building blocks such as amino acids or monosaccharides are strung together to form a polymeric chain, such as a peptide or polysaccharide. Some atoms, typically in the form of a water molecule, are discarded from each building block. And again, exactly like this setup here. So you connect these, and now you're going to remove this one here. So then these would be considered as a peptide residue or amino acid residue. So it's not actually formed of the full amino acids because you're losing some of the molecules, such as water. So it's a residue. And yeah, going back here, yes, or discarded from each building block, leaving only a quote residue of the building block that ends up in the finished product. But oftentimes you could just mention, without necessarily being as specific, saying it's residue, you could just say amino acids linked up. But to be more specific, you include residues because you account for all the little molecules that are lost during the process. So a residue might be one amino acid minus the discarded atoms in a polypeptide, or one monosaccharide in a starch molecule. Or this is a polysaccharide or oligosaccharide, just basically multiple saccharides. Going for multiple saccharides or carbohydrates and so on. A condensation reaction is the combination of two molecules to form a single molecule, usually with the loss of a small molecule such as water. So a protein sequence or protein primary structure is the linear sequence of amino acids in a peptide or protein, which is just a whole bunch of peptides. By convention, the primary structure of a protein is reported starting from the amino-terminal or N end to the carboxyl-terminal C end. So this would be the starting point, usually how it's referred. So this would be M, A, R, V, etc. So you would label it from these sides. And then there's a C terminus or end side on this side. Yes, very interesting stuff.